Welcome to Chapter 4. In Chapter 4, we're going to talk about the parts of the cell. So this chapter marks the rejoicing for many students as we leave the world of chemistry and get into some more serious biology. As usual, I have a list of learning objectives for my lectures. These are related to your study guide questions. So continue to use your study guide questions however they're working for you, answering them, just skimming through them, whatever works for you. But I do encourage you to start using them if you haven't been using them and to continue if you have been. On Blackboard, I have posted several videos. There are probably at least 5 or 6 video links there for you to get some more information on some of these things. 2 of them are called A Tour of the Animal and A Tour of the Plant Cell. Those are videos that are put out by our book publisher. They're not super exciting, but they're really pretty good animations and explanations of the parts of the cell. And then there are a couple of other outside links that you can get through YouTube that talk about cell theory, which is something that is really important in biology, but that our book doesn't really talk about too much. And then the endosymbiont theory that I will get to later when we talk about the mitochondria and chloroplast. And that one just tends to confuse students a little bit. So there is more information related to this chapter if you need it. I will not be testing you on section 4.1, information about the microscope, because we go over the microscope in the very first lab, but the information is there if you need it. So I said that we are done with chemistry for now. We will come back to a lot of these molecules that we talked about in chapter 3 and their structure. So we're not fully done with chemistry, but we are done with the chapters that focus on chemistry. So why did we talk about all this chemistry? Obviously we're a biology class, but our first 2 major chapters are chemistry. The reason that we talk about chemistry is that cells are made up of collections of molecules. So remember this idea of emergent properties, where the organization of something in a particular way leads to a new function. Cells and organelles begin to function when you have molecules in a particular form, in a particular structure, and it is the cell that we are most worried about, because the cell is our smallest collection of matter that's considered alive. So it is the smallest unit that is considered living. And atoms and molecules make up those cells. We are going to look at some of the different organelles. Almost all of them are made up of membranes, which are made up of mostly lipids, some other molecules as well, but mostly lipids. And lipids are really important in forming the boundary for those organelles and the boundaries for those cells. So we're actually going to talk very extensively about some of the molecules that make up these parts of the cells and the cells as a whole. And those atoms are important because they allow our cells to perform the functions that are required for life. It's not just chlorophyll and chloroplast, as we see here, but there are many other molecules. We have our DNA, we have our proteins, we have our carbohydrates. All of them have very specific functions in the cell, and the cell would not live without its full complement of those biological molecules. So let's go back a little ways, just a little bit of history here. So the history of the cell really begins with the invention of the microscope. So microscopes were invented in the 1590s, but the first cell, the first documented case of observing the cell, was not published until 1665. But this is still almost 400 years ago. So it's been a very long time. The first cell was observed by Robert Hooke. He built his own microscopes, and he decided to look at different things under the microscope. And one day he was looking at a piece of cork, and he saw that this cork had very specific ordered structures with what appeared to be a wall around it. And so he called them cells after the idea of rooms in a monastery, so little small rooms. And that name sort of stuck. A little bit later, a few years later, we have Anton von Leeuwenhoek. And he also was making his own magnifying lenses, and he really kind of observed anything and everything he could get his hands on. He observed the gunk in his teeth, he observed sperm, he observed all kinds of things, and he related the idea of these moving cells, he equated that to them being alive. And so he called them animalcules, these very little animals that were living. And in a way, he was right. He was looking at cells, which are indeed the smallest unit. So he really observed the first living cells and noticed, realized that they were living. So 1830s, jumping ahead a little bit, we have two men, a botanist named Matthias Schleiden and a zoologist named Theodor Schwann. They were not working together, but they kind of came up with the same ideas about the same time. So they observed that different parts of plants and animals are it should not say plants and animal cells. Different parts of plants and animals are made up of cells. So different parts are made up of different types of cells. And they recognized that these all had specific functions. They also theorized that cells were produced from other cells. So cells or life was not just spontaneously generating. So they were sort of the first to really put into words some of the characteristics of cells. Later, we have a few other individuals who were also stating that cells were made of cells, made from cells, not formed. So they didn't, again, they proposed that they weren't just appearing out of nowhere, but you actually had some sort of continuity. One cell begets another cell. In 1859, we have Louis Pasteur doing a lot of really important work. So he was a very famous chemist, and he gave us a lot of important things. The thing that he's probably most famous for is pasteurization. So the process of heating up usually a liquid to high enough temperatures that it doesn't change the liquid itself, but does kill any bacteria or other living things in it. So milk is pasteurized. He was actually looking at pasteurization of wine. But he did a lot of different experiments with bacteria and things like that. And so he was the first one to sort of show that spontaneous generation, or the idea that cells could come out of nothing, was really incorrect. So we have this experiment here that looks quite complicated, but it actually is not. So what he did was put non-sterile liquid, so something that contained living organisms, and poured it into a flask. He heated all of that liquid up, and then found that as long as living organisms could not get into the flask, nothing would be living in there. So nothing was spontaneously generating in this liquid where he had killed all of the cells. On the bottom, we have some different situations. We have a situation where the microorganisms could get into the flask and contaminate the liquid, and then we see that those microorganisms can grow in the liquid. So we only get growth in the presence of other living organisms. So this supports the idea that cells can't come out of nothing, and also that you require previous cells to make the next generation. So out of all of this, we get this cell theory. So the cell theory is made up of three very specific tenets. The first is that all organisms are made up of one or more cells. This tells us that the cell is the smallest unit of living organisms. So anything that is alive is made up of at least one cell. If it doesn't have an entire functioning cell, it's not alive. We also see that cells are the fundamental and structural unit of life. This is another way of saying that cells are the smallest unit. Nothing smaller than a cell. So an individual organelle cannot perform all of the duties required for living organisms. And then finally, all cells come from pre-existing cells. So you have to have a parent cell to produce another cell. There is a more modern cell theory that's a bit more complex. It takes into account some other things like converting energy and interacting with the environment, some of the other characteristics that we use to describe living organisms. But we're going to stick with the classic one. If you are interested, there's an article about cell theory and a little bit more on the history of the cell posted on Blackboard. So let's talk about cell size a little bit. Cells are really, really small. If you look at the right there, you can see this chart, a comparison chart that shows a variety of different types of cells. You see the human height over there. You see a picture of a woman. And then we see nerve cells, and some muscle cells are actually quite long. You can see them with the unaided eye. Then for reference, we have a chicken egg, frog eggs. Frog eggs are sort of getting close to the lower limit of something we can see with the eye. They're quite small, but we can still see them. And then we get down to a human egg, which is very large for a human cell. And then most plant and animal cells require the use of a light microscope. So this is the microscope that we're using in our lab. And so they're quite easy to observe on this microscope. We do it many times throughout our class. Getting much smaller than animal or plant cells can sometimes require the use of a different type of microscope. Most bacteria can easily be seen with a light microscope. The nucleus is the largest organelle in a cell. You can see those in a light microscope. If you remember from last week's lab, we looked at cheek cells. If you were able to do that and stain it, you saw the nucleus. There's a big purple stain in the middle. And then there are a couple organelles. Sometimes you can see the mitochondria, and you can definitely see the chloroplast with the light microscope. Below that, below a eukaryotic cell, anything smaller than that, you really need an electron microscope. And we don't use those at SWIC. They're quite expensive, but they do give you some really good resolution and allow you to see really small bacteria, even some viruses, ribosomes, things like that. And then as you get much smaller, you really start to run into the realm of something you can't see with a microscope, the individual molecules and atoms, things like that. So cells are often measured in something called a nanometer, which is a billionth of a meter, or a micrometer, which is slightly larger, which is a millionth of a meter. So that gives you a little bit of a indication as to the size that we're looking at. We see there plant and animal cells range from roughly 10 to 100 micrometers or micrometers, you can say it either way. So they're very, very tiny. Related to cell size, though, is the surface area to volume ratio. So this is really important for normal functioning of a cell. The cell size is actually limited by how large of a volume you have for the surface area. So if you think about a very large ball, say a kickball or something like that, you have a certain volume inside of it, and then you have the surface area around it. If you have a golf ball size, you have a much smaller volume for the amount of surface area you have. And this is really important because cells need to move things through the cytoplasm to the outsides of the cell. They also have to exchange things with their environment. And so the cell has to be small enough that the cellular products, waste products, things like that, can easily be moved and efficiently moved from wherever they are in the cell to the nearest edge. So if the surface area to volume ratio is too large, if the volume is too high for the available surface area, that limits the amount of metabolic activity that it can perform. The cell simply cannot keep up with the amount of energy it needs, the amount of proteins, the amount of molecules it needs to produce in order to keep up all of its processes if the volume is too big. Surface area, if there's not enough surface area for that volume, it limits waste removal. And as I said, it also limits any sort of gas exchange that cell is doing with the environment. If the volume is too large, the cell is producing too much waste, and there's not enough surface area to efficiently remove the waste or to bring in or release gases and other products throughout the cell, or across the cell membrane. And then I actually started with volume. Volume limits movement within the cell. So if the volume of the cell is too big, the cell cannot efficiently move things around in it. And we'll talk a little bit later about what kinds of things it's moving, but the cell needs to be able to move things within the cell. So all of that to say that a cell has to have a lot of surface area to less volume. So we say that this is a high surface area to volume ratio. This image here shows a surface area to volume ratio, and I'm sorry that I've got this, oh there it goes, I couldn't get the toolbar to move. So up here we have a 1 unit square cube. Next we have a 5 unit square cube. The first row shows us the total surface area. Underneath that we have the total volume, and then we have the surface area to volume ratio. So they're just dividing the surface area by the volume. So if we look at that 1 unit square cube, we see that we have a surface area to volume ratio of 6. So we'll just say that that is our reference point. Next to that we have that 5 unit squared cube, and if we look at the surface area to volume ratio, we see a 1.2. So this is much lower. Remember that I said that cells need to have a high surface area to volume ratio, so we're really looking for a larger number. So that small cube has a higher surface area to volume ratio. So cells can do a couple of things to to increase their surface area to volume ratio and to limit their volume. I'm going to come back to this picture in just a minute, but they can do a few things. They can make a bunch of smaller cells instead of a few larger cells. They can also make their cells long and thin or flat. So this spreads out their surface area and has thinner volume across the entire surface. Cells can also produce something called microvilli. So microvilli are folds in the cell membrane, and that just increases the surface area. So it's like fingers sticking out. If you look at your fingers, you have surface area on all sides, whereas if you ball up your fist, you don't have that surface area in between. And so all of these things increase the surface area to volume ratio. So if we look back at our this cube here that is a series of 5 1-by-1 cubes, you can see that you have surface area in between all of these surfaces. And if you add up all of the surface areas, you add up all of the volumes, and you take the ratio, we see that we get down to that surface area to volume ratio of 6. So again, a really high surface area to volume ratio compared to just one larger cell. If you remember from chapter 1, we talked about 2 types of cells. our two categories of cells are prokaryotic cells and eukaryotic cells. And there are some characteristics that all of these cells share. There are also a variety of things that make them very different, but all of these cells share some very similar properties. So the first is they are all bound by a plasma membrane. Plasma membrane is just a membrane that surrounds the cell. It's made mostly of lipids, and I'm not going to talk about it too much here because we talk about it extensively in Chapter 5. But for now, just recall that we talked about phospholipids in Chapter 3. They have the characteristic hydrophilic head with a hydrophobic tail, and that gives them some special properties that make them really good at being a membrane around the cell. Next, all cells have a cytoplasm and a cytosol. So the cytosol is a sort of a liquid matrix around the organelles. It's almost like a jelly that fills the space between the plasma membrane and the nucleus. The cytoplasm is everything within the cell. So anything that is inside of that phospholipid bilayer plasma membrane, all of the organelles, the nucleus, that's an organelle, all of the cytosol, anything that you find within the cell membrane is the cytoplasm. All cells contain chromosomes, so they all have some sort of genetic information, and all cells contain ribosomes. So I'm going to talk about both of those just a little bit later. Let's talk specifically about prokaryotic cells for a minute. Prokaryotic cells do not have a membrane-bound nucleus, and they do not have other membrane-bound organelles. But remember, they still have a cytoplasm, they still have cytosol, they still have a membrane around the outside, and of course they still have DNA and ribosomes. Although DNA is not isolated in a nucleus, it is concentrated in a region called the nucleoid. So it is sort of all compact together, and it still takes up a large volume of the cell. It's just not specifically isolated from the rest of the cell. And although the prokaryotic cells don't have organelles, they do have cytoplasm that has regions that are for specific functions. So a lot of bacteria and archaea they all will perform very similar functions to eukaryotic cells. They're just not isolated in plasma membranes. So you can see in our picture here, we've got the bacterial chromosome in the center. Inside, we have, it's sort of a darker yellow, the plasma membrane. Bacteria are often covered by a cell wall for support. They also have ribosomes in there. Those are the little dots. And then this picture is also showing flagella and fimbriae. Those are appendages on the cell for movement and communication with the environment, with other cells, or sort of sensing the environment. Eukaryotic cells are much more complicated, and they're more complicated due to the presence of organelles. So organelles are really important because they each provide a specific function, and they allow the eukaryotic cells to do more jobs. So in the next video, we're going to talk about all of these different organelles and what they do. Most of them are the same between the plant and the animal cell. So we will talk about all of them, and then we'll talk about some that are specific to the plant cell and just a few other characteristics of the plant cell in the next video.